Hello everyone, it is Mitchell. I am a maths and science teacher, and I thought that today we'd go through a GCSE biology paper. So the reason why I picked biology to do the first is because that was the one that was most requested. So I did a biology, chemistry and physics paper last year, and they were pretty well received, but the biology one was the one that had the most requests for another top video. So instead of doing a paper 2, we're going to do a paper 1. I'm going to explain the common misconceptions, and we're going to answer all of the questions in detail, and I'll explain where you go wrong. I'll explain the mark scheme to you as well. So without further ado, let's go on to the first question. This question is all about cells and transport. Complete table 1. So we have a name of cell, and we have a function of cell part, and we have to just say. So the name of the cell part, what contains genetic information? That would be the nucleus. So it isn't DNA technically, because that's not a part of the cell. That's just what is inside the cell in and of itself. The mitochondria, all you have to do is write respiration. However, if you say aerobic respiration, that is also fine. Manufacturing ATP is also fine. The only thing you can't say is anaerobic respiration, because that is wrong. So anything along the lines of producing ATP, respiration, aerobic respiration will get you the marks. And lastly, we have controls the movement of substances into and out of the cell. That is, of course, the cell membrane. Again, that's mostly a fact thing. So what's the best way to memorize this? I would recommend using flashcards, just for the ones that you're unsure of. So go back and think about what parts of the cell give you a bit of trouble. Just write those on flashcards. There's no point in writing flashcards for all of the parts, because you already know most of them or some of them. Do the ones that you don't know. Cells in potatoes are plant cells. Cells in potatoes do not contain chloroplasts. What is the function of chloroplasts? So of course, in this case, they're talking about photosynthesis, but what do you need to actually write in order to get the marks? Well, all you need to do is write photosynthesis. As long as they see the word photosynthesis, you're fine. If you want, you can say that it's the site of photosynthesis, but that is, unnecessary. You can just write photosynthesis. And this is something I recommend to people, is in your GCSE science exams, physics, bio and chem, you can write in short form. You can write in bullet points. You can just write single word answers. You don't need to write in full sentences. Even though it says quality of written communication, that doesn't mean grammar and spelling. You used to have to write in full sentences, but not anymore. You could also write something like to produce sugar or glucose, because that's what photosynthesis does. And you can also say absorbs sunlight. You can't just say, oh, it contains chlorophyll. That's not the function. That's just a property it has, an adaptation it has to help it with its function. So the actual function is photosynthesis, or produce glucose, or produce sugar, or absorb sunlight. One of those there. Name one type of cell in a potato plant that does not contain chloroplasts. Well, you could say anything along the lines of a root hair cell. Now, by the way, one thing, just taking myself out of the question for a second, whenever I come across this, think about what the function is and think about where you wouldn't need it. So with a plant, well, chloroplasts are for photosynthesis. So where would I not need photosynthesis? Well, where it's dark. Where is it dark? Underground. Or inside the plant itself. So underground is probably the most common one, root hair cell. But any cells that are on the inside of a plant only, such as the xylem or the phloem, will also not have chloroplasts because they're on the inside of the plant. How are they going to get sunlight? You could also say epidermis or the meristem as well. So just keep that in mind. Think about the function and think about where it will not be needed or will be needed. That goes for even like animal cells as well. Like they might say something like, where are epithelial cells found in an elephant or something? Well, think about what they do and where you'd need that. And that's pretty much it. That's what evolution has done. So that's all we need to do. And we have an investigation. The effects of salt concentration on pieces of potato. So they cut 3 pieces of potato of the same size. So same size, it means it's a control variable because they're controlling it. They record the mass. They add this solution, a salt solution, into a beaker. They place a potato in it. They remove the potato piece and dry the surface. Record the mass of each potato piece and repeat the steps using different concentrations of salt. So they're trying to draw water out of potato, basically. Now, independent variables. This is something that people struggle with in biochem and physics, so this is useful for all of them. And I've got a really easy way to remember it. An independent variable is the thing that you change or control. So it's the thing that you're changing. So if I read through this, what are they changing? Well, it says repeat using different concentrations of salt. They're controlling it, which means concentration of salt solution, because they're the ones, the person doing this, so this student is the one changing that. They are not changing the mass of the potato. The mass of the potato is changing as a result of the salt solution. But the thing that they're directly changing is the concentration. Now, that means the dependent variable is the one that depends on the independent variable. So it's the thing, and here's the easiest way I can think of to remember it, is the dependent variable is always the one that you measure 100% of the time. You measure your dependent variable. So, if you're investigating how mass affects velocity, for example, velocity is what you're measuring. So whatever you measure is the dependent variable always, and therefore the independent variable should be the other thing. There are only two things that will change in any experiment: the independent and the dependent variable. So if you know what the dependent is, you immediately know what the independent is. So you can think of it as both ways. The independent is one that you yourself are changing. The dependent is the one you're measuring, always. What are control variables? Every other variable inside an experiment is something you're keeping the same. So for example, cutting the piece of potato of the same size, that means the size of the potato is control variable. Also the volume of the salt solution, so the concentration changes but not the actual volume itself. 30 minutes of the time it's left in there, that's also a control variable. 1.5, why did the student dry the surface of each potato piece with a paper towel in step 5? So we need to really think carefully here. So step 5, after 30 minutes they remove the potato piece and dry it. What are they doing afterwards? They're recording the mass. Now if you think about, I'm hoping you've done this experiment yourself, but when you cut these potato pieces off, they're very small. In fact, they only probably weigh between 10 and 30 grams, depending on how big you cut them. That's a very small number. If we leave water on the surface, we are adding on the mass of the water. And again, that's going to be different every time we remove the potato piece, because that's just down to random luck how wet the potato is after we've taken it out. So that's why. There's a bunch of ways to write this. You can say something like, allow the removal of excess water. You can say to make sure only the mass of potato is recorded. Potato is recorded. You could say if the water was left on the or the solution was left on the surface, we're measuring the mass of that. Anything that implies that the mass of the solution is going to affect the recording works. So essentially we just want the potato to be weighed, nothing else. So we have to remove all excess stuff, for want of a better term. The student calculated the percentage change in mass of each potato piece. For one potato piece, there was a starting mass of 2.5, ending was 2.7. Calculate the percentage increase. Increase in mass, well, by the way, this is just GCSE maths. It's percentage change. So the increase in mass is just going to be the difference in these two numbers. So that's 0.2 grams. So 0.2 over the starting mass, which is 2.5, times 100. I could probably do this in my head. So that's 20 over 2.5. And then, you know what, let's just use a calculator. Open up the calculator. 20 divided by 2.5 is 8%. Ah man, I really could have done that, couldn't I? 8%. Nothing too spectacular about that in my opinion. After getting all these results, they wanted to calculate the mean percentage change in mass. So what they've probably done is repeated it a bunch of times for each concentration and then done the calculation. So we need to complete the figure. We need to label the x-axis, use a suitable scale, plot the data, draw a line of best fit, all of those wonderful things. So this is just plotting graphs. So the x-axis, so if the y-axis is the mean percentage change, what would the x-axis be? Well, simply put, it's just the other thing. So the concentration of salt solution. So concentration of salt solution. I would recommend writing out the whole damn thing, because, they can't drop any marks for that, simply put. And what is this going up in? It's going from 0.0 to 0.4. So we could probably afford to go up in like 0.05. So 0.51, 1.52, 2.53, 3.54. I think that's reasonable. So 0.05 there, 0.1, 0.15, 0.2, 0.25, 0.3, riveting I know, and 0.4. I believe it only went up to 0.4, but let's double check. Yeah. And then all we need to do is plot it. So 9.8 for the first one. So this goes up in 0.2s, yeah, 2, 4, 6, 8. So at 0 we're at 9.8. 0.1 we're at 9.5. And again, this is just plotting graphs. It's not the end of the world, I'm hoping. Wait, hold on. Did I read that right? Yeah, 0.1 is 9.5. That was close. 9.245 is right there. 0.2 is 7. Cool. 0.3 is 0.4. It's not 0.4, it's 4 even. I promise you I can read. No, it's not, it's 0.4. Never mind. I should have trusted myself. That'll be all the way down here. And 0.4 is negative 1.424. So line of best fit, you want something that kind of goes through most of the points. So I would probably argue, again, I could use a ruler, but I would argue something along the lines of that works. So another thing that people like to do, and those people being me, is I count, if there's 2 below the line, there should be 2 above the line. That's the kind of rule I use. So if there's 6 points, there should be 3 below and 3 above, unless it goes through one of the points. But something along those lines. You don't need to be super mega ultra accurate with it, but yeah, you just need to have that kind of rough shape there, and they're not too picky about it either. What concentration of salt solution was equal to the concentration of the solution inside the potato peas? Interesting. So what would that mean? Well, that would be when there is no, okay, going back to the experiment, why does the mass change? Well, simply put, because of the difference in salt concentration, there is a difference in concentration of water inside and outside the potato, which means osmosis happens. So let's say inside there is a concentration of 1 mole, outside there's 0.4, then what's going to happen is water will diffuse out, or do osmosis out of the potato until you have a balance, let's say of 0.7 and 0.7. So, how can we tell when there is no difference? That will be when the change in mass is 0, because there is an equal amount inside and outside of the potato. That means there's no need for osmosis to happen. Osmosis won't happen, which means the mass will stay exactly the same, assuming you've dried it off properly. So what we need to do is look for when the mean percentage change is 0. So when's that? It's when it crosses the y-axis, which looks to be about here. So if I zoom in, it goes up in 0.01, so that I'm going to say 0.36. And they do allow you a fairly big range for this question. But also they need to see some evidence you've used your graph. So I would probably try and get another color and like do a really big dot just to show everyone, hey, I am using the actual graph that we've just drawn. And lastly, explain why the potato pieces in the 0.4 salt solution decreased in mass. Well, it's basically what I just said. So I'm actually going to type this out. So if we move this over, and again, using bullet points is probably the best way to do this, because you avoid writing excess stuff. You just want the individual points. So you get marks for writing each of these points. I'm just thinking about the best order. I think we should say the difference in concentration first. So because there is a difference in concentration of salt inside the potato compared to outside the potato, water will move out of the potato through osmosis. Those are your 3 marks. That's literally all you have to write. And again, hopefully you can see the logic in me using bullet points. I use bullet points because it avoids waffle. Waffling will... I see people with 3-mark questions filling up the entire thing and writing down like 4 or 5 lines worth of stuff. It's a waste of time. A, if you contradict yourself, you will not get any marks. And B, you might only get 1 mark. Think about the key things that examiners want. What do they want? They want the name of the process, so write osmosis somewhere. Then they want you to say, well, what is that process? Well, it's when water moves, in this case, out of the potato. And what's the effect of that? Or why does that happen? Because there's a difference in concentration of salt. That's what they're looking for. They're looking for names, explaining the processes and why those processes happen. Or what effect do they have, depending on the question. So this is also why I recommend for GCSE Science doing every single paper multiple times. Because realistically, what you're really trying to do is memorize where the marks are given. That's all you really need. Question 2 kind of continues with cells. So plant and fungal cells are similar in structure. So you don't actually need to know much about fungal cells. Not that much, not compared to eukaryotic and prokaryotic cells and plant cells. So we have some kind of fungal cell here. We have a big white mark, we have a dark spot, and then we have some other stuff. And it says, name 1 structure that's present in both plant and fungal cells, but not in animal cells. So again, part of the spec does say that you need to be able to be given a random cell, whether that's from a different kingdom, so whether it's from plant, animal, fungal, etc., protists, whatever, or from a different specialized cell that you haven't spoken about, maybe like a cell that only lions have, for example. And you should be able to describe it. So if I have a look at this, what are these things? Well, this black dot is the nucleus. This background is cytoplasm. Again, even if I've never seen this cell before. This is a cell membrane, that's a cell wall. And this, well, what is this? Well, if you look at what it's described as, it's a big white empty space. What is a big empty space? It's a vacuole. It's a permanent vacuole. Whoops. So again, I've just labelled every single part of that cell, even though I've never seen it before. But think about it. Well, these outer layers are obviously the membrane in the cell wall. So now think about what ones are in plant cells as well, but not in animal cells. Well, I can't say nucleus, because that's in every cell. I can't say cytoplasm, because that's in every cell. I can't say cell membrane, because that's in every cell. But I could say cell wall. And I also could say the permanent vacuole. So again, just from looking at it, you should be able to kind of roughly tell what these different things are in the cell. Which disease is caused by fungus? This is just a memory game, really. Malaria used to be considered a virus, but now it's called a protist. Measles is a virus. Gonorrhea, STI. Rose black spot is indeed the fungal one. That's just memory. Again, that's a really good place for flashcards. If you already know certain ones, like let's say you know malaria is caused by protists, then don't write the flashcard for it. But if you got stuck on gonorrhea, then write the flashcard for it. That's all I would say. And that way you have fewer flashcards. I know some students that have 100 plus flashcards. Well, that means you're only going to go through each one once in 100 times. Whereas if you narrow that down to 50, you're going to see each one twice as much. It's a lot better. A fungal cell divides once every 90 minutes. How many times would this fungal cell divide in 24 hours? This is a math question, pretty much. So 90 minutes, so how would I do it? You can either convert hours into minutes or minutes into hours. So I know, for example, 90 minutes is one and a half hours. How could I know that? Well, think about it. How many minutes are in an hour? 60, so 90 divided by 60 gives me 1.5. If you want, you could change hours into minutes, you get the same answer, it don't make a difference. And then you're just saying, how many times would it divide? Well, in 24 hours, what they're asking you is, in 24 hours, how many 1.5s are there? So how many times does 1.5 happen in 2.4? Well, 1.5, 3, 4.5, blah, 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 blah. We can just use our good old-fashioned calculator for that. Divide by 1.5 gives us an answer of 16 times. Whoop-de-doo. which means it duplicates 16 times in 24 hours, which is kind of terrifying, but that is what it is. Some types of fungal cell are grown to produce high protein food. Very true. The high protein food can be used to make meat-free burgers. Where is protein digested in the human digestive system? Tick 1 box. Cool. Well, again, probably the best way to do this is to label a human digestive system and label it not just in terms of names, but have it, so for example, your diagram's this big, have it on a page that's that big, and you draw a line, so stomach, what happens in the stomach? What, not proteins, what are they called? Enzymes, there we go. What enzymes are present in that, and then thus what is actually digested? And do that the same with the mouth, the salivary glands, the large intestine. That's the best thing you can do. So have a diagram, then draw out and have lines of text, just quick, brief things. Okay, in the stomach, what is the stomach? What is important about it? Oh, okay, it has a pH of this. It digests this. It produces these enzymes. And do that with the mouth as well, and with the intestine. Just write all that out. And the best way to do that is visually. You can print them online, but nothing beats doing it yourself. So if you want, you can have it online and then write it out yourself. But again, don't copy. The idea is that you're doing it yourself. When you make something, you remember more of it. But in this case, it is indeed the stomach. Protease in my mind is always really like hot. I don't know why. So that's how I remember the stomach, because it has the lowest pH. It's the most acidic. I have no idea why that's associated in my mind, but it is. Which chemical could be used to test if the burgers contain protein? So basically, which one tests the contents of proteins or amino acids? Burette reagent. Again, good old-fashioned flashcards would probably be best. Okay, now we're talking. We have some information about burgers made out of meat and meat-free burgers. So we have protein, we have fiber, okay, fat, okay, carbohydrates. And I do recommend reading through these. And then cholesterol. Damn, 0? Okay, fair enough. Evaluate the use of burgers made from meat compared with meat-free burgers in providing humans with a healthy, balanced diet. Okay, so what the hell does evaluate mean in terms of science? Evaluate in English, maths, and science means 3 different things. Evaluate in maths, for example, means you're going to get an answer, as in there is a fundamentally correct and wrong answer. With English and science, you're evaluating, i.e., you're comparing both sides. So, evaluate the use of burgers. Well, what we're going to do is we're going to say, well, we're going to compare the protein. Oh, well, burgers made from meat are higher in protein than meat-free burgers, which means, you'd have to eat more meat-free burgers in order to get the same amount of protein in your diet. Cool. Fiber. Oh, well, meat-free burgers have higher fiber. I'm not even kidding. You do that, you get 6 marks. That's it. But you do need to link it. So why is protein important? Why is fiber important? Why is fat important? Why is carbohydrates important? Why are cholesterol important? 6 marks, a lot of stuff. It does say and your own knowledge, which is fairly interesting. So, if I were comparing it, what would I like to do? Well, literally, again, bullet points. So if I say meat burgers contain more protein, why is that useful? Well, I would say because, well, why is it useful? For growth. For growth and repair. Boom, there you go, 1 mark done. Next, fiber. Meat-free burgers contain more fiber. I'm not even kidding, this is all you need to do and you get 6 marks. I'm literally just reading what's in the table. But what is fiber used for? Aids in digestion. Aids in digestion. There you go, 2 marks. Next, fats. Well, I mean, this way you could consider it positive or negative. But again, we don't have to say positive or negative. We just say meat burgers contain more fats. And why? What is the problem or good side of that? Well, what does fats remind you of? Well, bad things. Okay, cool. What bad things? Well, could be obesity. Oh my god, I can't spell obesity. There it is. The I and the S got swapped around. Okay, thank god. Okay, what else could it be? Again, that gives me the mark, but what else could it be? Well, it could be heart attack, stroke, coronary heart disease, angina, diabetes, may lead to needing a stent in order to treat the coronary heart disease. Any of those things would work. But you only need one of them. So only put one of them. Again, waffling is not good. This will get me full marks in an exam. Carbohydrates, which one has more? They contain about the same. I mean, you can say technically the meat burgers have more carbohydrates, but I'm going to say they're roughly the same. Both burgers have approximately the same amount of carbohydrates. In fact, that's the longest sentence I've written, so I might want to shorten that and just say both burgers have same carbohydrates. Talk like a caveman, it's fine. Cavemen are awesome. But what are carbohydrates used for in the body? To provide energy. Wonderful. And lastly, cholesterol. Meat burgers contain far more. You can just say more, but I like to exaggerate. I'm a very annoying person. Well, why is that bad? Well, what's cholesterol bad for? Well, could be anything. Again, could lead to coronary heart disease, could lead to a heart attack, could lead to taking statins, could be anything. By the way, something that's really cool that I like is you don't have to write coronary heart disease, you can write CHD, which is a lot better. May lead to heart attack. There you go, 6 marks. But yeah, CHD, coronary heart disease, is also accepted in the mark scheme, which I am very, very happy about. That's it. You don't need to write pages of stuff, but the worst thing I see is when people write paragraphs and they get like 4 out of 6 because they haven't written about all of them. You have to read each one, you have to compare each one, you have to evaluate. You have to. So just do that in as few words as humanly possible. Student prepared some onion cells. The student viewed the onion cells using a light microscope. This is the method used. Cut an onion into pieces using a sharp knife. Peel off a thin layer of epidermis. Place on the microscope slide. Yep, cool. Add iodine solution. So that way you can actually see the cells themselves because they're normally transparent. Slowly lower the cover slip at an angle onto the onion epidermis. That way you don't crush it. Instead you're layering it on. Place slide on the stage. Okay. Shows a risk assessment. Okie dokie. Iodine solution is an irritant. Indeed. May cause an allergic reaction or skin rash. Plan to minimize it. Wear gloves. Yeah, not too tricky. Some other things you could say. Wash skin immediately if there is contact and wipe down any spills. That's the best thing. Sharp knife risk. Well, gee, I wonder. You may cut yourself. It's easier than it looks, I promise you. So what do you want to do? Well, there's loads of things you can do. The number one thing, whenever you, by the way, this is just life advice, whenever you use something sharp, like a standing knife or anything like that, cut away from you. So don't cut towards you, cut away. I know that might sound obvious, but you would not believe the things I've seen, even in a university physics lab, when someone had to cut something. And yeah, just cut away from the body. You can also use a chopping board. The reason why this is important is your lab surfaces are actually a bit slippery. Same with the kitchen surface, by the way. I've seen people try and cut on a kitchen surface. What happens is they put pressure on it and onions around, or potatoes, whatever you're using, and they slip. And when you slip with a knife, your finger's the only thing that's going to be cut. Keep fingers away from the blade. Yeah, that's probably it. Yeah, that would probably be the only thing. In terms of, there is one thing that I heard someone say once, which was avoid using a blunt knife, and I get what you're saying. If you use a blunt knife, you have to put more pressure on, and so it's more dangerous. Not accepted, unfortunately. Give a reason for each of the following steps. A thin layer of onion epidermis is used. Well, why does it have to be thin? So you can see through it. There's a few different things you could say. So you can either say, so light can penetrate. Because remember, we want it to be like slightly transparent. Light can penetrate. You could also say to help see individual cells. Why is the iodine solution used? Well, so you can see parts of the cells. So it's to stain. There's no E in stain. To stain the cells so you can see it more easily. Why is it lowered at an angle? To prevent air bubbles. basically what happens when you put it on just flatly is you end up crushing the onion layer, which can release air, which means you have these little pockets of air which are really bad. If you do it like that, it works a lot better. That's also why, I don't know if you guys have to use sticky back plastic anymore for your books, but again, if you put it on flat, you get air bubbles, whereas if you layer it on, it looks a lot nicer. That's just, again, if any of you do art or anything art related, you probably know to do that as well. Next, here's what the students saw. So you see these individual cells, these are the cell walls, those are the nuclei. Fancy. The length of the cell, which is this thing here, is 4.8 centimeters. Calculate the real length. Well, it's a 400 times zoom, right? So this line here, this image, is 400 times bigger than it should be. So we need to do 4.8 divided by 400. Now it says give your answer in micrometers. So what might be, there's a few different ways you could do this. Let's just do it the normal way. 4.8 divided by 400. Well, if I divide it by 4 first, I get 120. But then I move it 3 decimal places, so it's 0.012 centimeters, which, if you remember, there are 1,000 micrometers in a centimeter, so you're moving it 3 places to the left, which makes it 12 micrometers. So remember, that is 1,000 in 1 centimeter. There's 1,000,000 in a meter, for example, so that's why I ummed-and-ahed a little bit. That's it for 5 marks. Wow, I love biology. In terms of those of you wondering, like, what about the equation? Well, yeah, the equation is that the magnification is the size of the image over the size of the real object. Size, image, over size of object, right? And then you may have to rearrange this and do all of that fun stuff, you know, so you're rearranging it for the size of the object, yada, yada, yada. But if you notice, when you rearrange it, you get the same thing. But I don't like that. I like to think about what the hell magnification is. Well, think about it. If I zoom in on my phone 5 times, that means the image is 5 times bigger. That logically makes sense. So in this case, this image is 400 times bigger. That's what times 400 means. It's a scale factor. So then why wouldn't I just divide by 400? I don't need fancy equations. Like, it's a magnification. It makes things 400 times bigger, so just divide it by 400 to get back to the original. Okay? Anyways, yeah. 5 marks in one or 2 lines. Pretty good. Pretty good. So, the student then draws it. This is a better drawing than mine. But give 2 ways the student could improve the drawing. A billion different things you could do. Include magnification. That's a pretty big one. So when you draw this, can you tell how big it is? Like, for all you know, this is how big the cells are actually in real life. But how do you know if it's 10 times bigger, 50 times bigger, 1,000,000 times bigger? You don't know. I would argue, so these gaps are kind of annoying. So if you notice, there's gaps between lines, so no gaps between lines. Between lines. What else? Well, if you look at the actual image, he hasn't drawn all of the cells, so he should draw all of the cells. He should use, what else could he say? Draw the correct shape. So this is a weird shape that doesn't exist here. So that might be a good one. Label the cells. Don't have gaps between the cells. Anything along those lines. Onion cells can be seen using an electron microscope. Give 2 ways it would look different when using an electron microscope. Well, the big one is that it will look bigger. So more magnified. More big or magnified. Both of those work. Again, the most important piece of advice I've ever been given in life is keep it simple, stupid. So what that means is, what's the most obvious? Well, a light microscope versus electron microscope. If you've been listening in your science classes, why do we even need electron microscopes? Because we need big magnification. We want to make things bigger, make bigger. So make big. It's going to be bigger. Shocking. So what does that mean when we magnify more? Well, that means there'll be more detail. You can also say more cell structures. So you might be able to see things like ribosomes, as a random one that I just picked out. But you'll be able to see more stuff. And that's it for question 3. Pretty good question in my opinion. Now we're getting on to defense responses and maybe even some adaptation stuff if I can predict the question a little bit. But we have defense responses, so immune system, stuff like that. Table 5 shows some plant defenses. Identify whether each defense is chemical or physical. Okay. So what's the difference? This is going to sound really stupid, but chemical is produced by chemicals. So anything that is spicy, poisonous, venomous, secretions, anything along these lines, like mucus, they're all chemical. Physical refers to something physically happening. So for example, the thick waxy layer is a physical thing. So it's physical. Poisonous, well, poison is produced by chemicals, so it's chemical. Bark that falls off, well, it falls off, right? That means it's physical. Chemical is literally only if something's really spicy, poisonous, venomous, mucus, something along those lines. That's probably the best way. Physical is literally when something's physically happening to the structure or the shape of the plant, animal, whatever it is. Mimicry is a mechanical adaptation seen in both plants and animals. So you have hornets and a hornet moth. So they're obviously two different species, but the moth looks like a hornet. Hornets are insects that sting other animals and cause pain. So that's a chemical response because the chemical inside the sting is what actually causes the pain. Hornet moths do not sting other animals. It's just how mimicry helps the hornet moth survive. God. Well, hornets are dangerous, right? Moths aren't. But if you look dangerous, predators will stay away from you. That's it. So predators tricked by colouring. And avoid the moth. That's it, really. Because hornets are scary, this thing wants to look like a hornet, so that way it's, people are scared of it, even if they don't have to be, which is amazing. Adult hornet moths lay eggs that hatch into larvae. Figure 6 shows the larvae of a hornet moth. Adorable little things, aren't they? They can be found living inside the roots of trees. Okay, use the tree roots as a source of food, cause damage to the tree roots. Explain why a tree might die if the roots are damaged. Okie dokie, 6 marker again. So, again, the best way to think about this is, why might a tree die if the root trees are damaged? Well, what do roots do? Well, they absorb nutrients and water from the soil. So, the best way to think about this is think of the extreme. Imagine the roots are gone. No water, no minerals, dead. That's the best way to explain it. So, 6 marks though, we can't just say no minerals dead, because of course not. So, there'd be less absorption of water. Okay, cool. Why is that bad? Well, that means there'll be a lower rate of photosynthesis. Okie dokie. By the way, what I might do is actually come back to this later, this next bit. The reason for this is you want to make sure you mention all of the causes. So that is quite important. So you might want to also say something like less absorption of minerals / ions. And why is that bad? Well, it means it will... there's loads of things, right? So if it's nitrates, fewer nitrates means less protein for growth / repair. And you can imagine that's quite bad then, because if the roots are damaged, but you don't have materials to repair them, then they're going to stay damaged forever, and they won't ever be able to repair themselves. So that's pretty bad. What else? So I'm just going to do one for each, and then come back and maybe add something more if I realise I don't have enough points. That's my approach to this. So in an exam, obviously it's harder for you to do because you can't edit stuff like this. So I might write this and then leave gaps in my writing. So the way I'm actually writing is more like this. And then I might come back and add in extra lines if I feel that I need to. That would be my advice for you. It could also damage the phloem or xylem, for example. Damage phloem / xylem. And why is that bad? Well, that means there's less transport of water and minerals / food which leads to, plant cells not getting enough minerals to function. So you could give an example there. For example, magnesium helps in photosynthesis. So if magnesium isn't getting to the leaves, then no photosynthesis, which is bad. So you can even go back now and just write this. A lower rate of photosynthesis. Okay, cool. What else? Which means there's less glucose. Which means there's less energy released. So count out the points. Let's say 1, 2, 3, 4, 5, 6. There's 6 points, so I'm gonna stop. But you can keep adding more and more points until you get to 6. So again, these 3, 4, 5, and 6. Just get 6 points and you're done. That is my number one piece of advice. There's no point in writing pages and pages and pages of stuff. The larvae of the hornet moth form when fertilized eggs divide by mitosis. Describe how mitosis produces two genetically identical cells. This, again, is such a good question because it is a standard 4-mark question. You just write the 4 marks, the 4 bullet points every single time. And you always get the 4 marks. There's no questions about it. Probably one of my favourite questions because of that. Again, my favourite questions now are not interesting ones. It's just ones you can easily get marks on. But whatever. So these are the 4 bullet points that I use. But if you have your own 4 bullet points, that's completely fine as well. But again, you just want 4 bullet points that gets you the 4 marks. 1 mark per bullet point. That way, even if you forget 1, you're still getting 3. You know what I mean? So, what happens first? Well, the DNA is duplicated. Oops. Cool. You can say copied or doubled or replicate, whatever you want. That's fine. Okay, that's 1 mark done. What happens next? The chromosomes are pulled apart. You can say separated. What happens next? The cytoplasm divides into 2. And then the last thing is, that's it. The set of chromosomes in both cells is identical. There are 6-mark answers, so some of you that may have seen the 6-mark answer where you have to go into a bit more detail. So make sure you memorize the 4-mark version and the 6-mark version, and that's it. So again, think about it. DNA duplicates, chromosomes pulled apart, the cytoplasm divides down the middle. There are now 2 identical cells. Done. The cells which are first formed from the fertilized eggs of the hornet moth are stem cells. Name the process by which the stem cells form specialized cells. It is differentiation. Differentiation. That's it for question 4. That was okay. Yet more plant-related stuff. I love it. Water and carbon dioxide exchanged between leaves and the atmosphere through pores called stomata. Name the cells controlling the opening and closing of the stomata. The stomata is like a gate. Who used to control how gates work? They were called guards. So these are guard cells. What defends the entrance to something? Guards. So guard cell. Something along those lines to help you remember it. It's just a memory game. Sorry. Water moves through a plant in the transpiration stream. Describe 2 differences between the transpiration stream and translocation. Okay, well, the first one is transpiration is xylem, translocation is phloem. So transpiration uses xylem, translocation uses phloem. That's one difference. Well, again, so basically, by the way, the difference between this transpiration and translocation. Transpiration is movement of water and ions. Translocation is movement of heavier stuff like food. So that's another difference. Is transpiration moves water and ions, whereas translocation moves sugars. The other thing that should be noted is the actual direction as well. Transpiration is always up, translocation is always down. So those are three differences right there. Transpiration is always water, translocation is sugar, transpiration is up, translocation is down, transpiration is xylem, translocation is phloem. That's it. Which environmental conditions would cause the rate of transpiration, so that is the loss of water through the leaves, to be greatest? Well, you want something that's hot and you want it with a low humidity. So why? Well, first of all, the warm should hopefully make sense. When things are warm, water evaporates. Cool. By the way, transpiration is just evaporation. Okay. Low humidity, why? Well, that means the concentration of water in the air is low, which means osmosis, the water wants to leave. So it's going to. So that's another way to think about it. And what is next? We have a figure. So going on to this question, we have figure 7 shows information about the mean width of the stomata in a plant. Changes in the mean width of the stomata in normal conditions are an advantage to the plant. Explain how. So this is simply talking about the reason why the stomata opens and closes in the first place. And why is that? Well, at night, let's start with at night. I think that's the best place to start. So at night, what's the problem? So why bother closing? And I genuinely believe that starting at night makes most sense because that's when these things close. Why do we need to close? Well, at night, the stomata are closed to prevent loss of water. That's one mark. Because there is no light for photosynthesis. So because there's no light for photosynthesis, there is no need to have the stomata open because we're not converting carbon dioxide into oxygen. So then why lose the water? If the stomata are open, you're not actually doing photosynthesis and you're losing water. With the stomata closed, you're not losing water and you're not doing photosynthesis. It's simply an efficiency thing. However, at, let's say, midday or during the day, there is maximum light intensity for photosynthesis. Obviously, because the sun is at its highest point in the sky, depending on what time of year it is. It's going to be at its peak at around midday. Therefore, stomata are open to maximize carbon dioxide intake for photosynthesis. So again, in the first case, when the stomata are closed at night, we don't need carbon dioxide because we're not actually doing photosynthesis. So we don't need to take in carbon dioxide. However, during the day, we need to open it to maximize this carbon dioxide intake. Now, this does mean that there is a lot more loss of water during the day. But that's fine because we're still doing photosynthesis. It has to be open at some point. Having open at night is like having a leaky tap, right? If you don't bother turning the tap off after you finished using it, you're just wasting water. Even though you do need the water to I don't know, wash dishes or whatever. So keep that in mind. And then for the next part, it's very similar to the previous one. However, it says the changes in the mean width of stomata in low atmospheric carbon dioxide are different from the changes in normal conditions. So again, in normal conditions means that when there's a normal amount of carbon dioxide in the air, it will be closed at night, open during the day. However, when you have low atmospheric carbon dioxide, what is the difference? Well, the difference is that the plant desperately needs carbon dioxide in the same way we need oxygen. So what it does is the stomata opens as wide as possible. So stomata opens wider. And this is like trying to take in a deep breath to get as much oxygen as possible. And for more time. So if you were in an oxygen-deprived room, for example, your breath will naturally get deeper and deeper because your body needs oxygen. In the same way, the plant stomatas will open wider and wider and for longer in order to take in more carbon dioxide. It works in virtually the same way human beings do when we need oxygen. So even if you couldn't tell there was low oxygen in the room you're in, you would start breathing deeper and for longer to take in as much oxygen as there is. So, why? It says explain the difference. We've stated the difference for 1 mark. Now we need to explain said difference. Well, The reason for this is it allows the plant, so to allow the plant to take more carbon dioxide in for photosynthesis. That's the reason. And again, the exact same reason why we breathe deeper when we need oxygen. Same kind of principle. Here's another style of question they can give you. So here we have a table of different organisms with a bunch of surface areas and volumes and the surface area to volume ratio. And all we have to do is calculate the value x, so basically calculate the surface area to volume ratio of organism C. Now what I really like about this is if you're unsure of what to divide what by, you can cheat. Let's say, for example, this bottom one, if I thought we had to do surface area to volume, so I thought, okay, let's do surface area divided by volume, I can check if that's the right answer. So 6.12 times 10 times 0, and I get approximately 3. So I know I'm meant to divide surface area by volume. If I did this the wrong way round, I would get 0.3, which I can see is not right. So if you're unsure in questions like this and they've given you a table, use some problem solving. Divide one number by the other, see if it's right. If not, divide the number by the other number. If that's not right, times them together. Try a bunch of different stuff to try and get it right. So it says give your answer to the nearest whole number. So we're doing surface area divided by volume because we've just double checked that that's what they did for the rest of the table, if that wasn't already clear to you. And we should get, well, actually I don't know what we should get, so let's quickly work it out. We get 73.7777. Okay, to the nearest whole number, that is 74 to 1. So x equals 74. So if you'd like, you can write out the calculation you did just to make sure you get some method marks. But again, we have got the right value, so we are all good in the hood. Nice and easy. I really like the calculations in biology because they're not that long and they're worth a fair few marks. 3 marks for that is pretty good. For part 2, it is a simple state question. What is the relationship between the size of an organism and its surface area to volume ratio? It doesn't say anything about explaining, which is fine because that's a mathematical reason, not a scientific reason anyway. So the size of an organism, what would the size be? Well, if we look at this diagram, at this table here, there's a few ways you can see this. 1, think about what size is. When I think of size, if I think of a 3D shape, when I think of size, I do think volume. So if something's bigger, it takes up more space. What is the mathematical term for the space a 3D object takes? It's the volume. The other way you can think of it is look at the table. There's only 2 numbers here which matter because it says here, don't forget, surface area to volume ratio. So this side, we know it's not this side that refers to size. We know it has to be one of these two. Now, you could kind of use either one because both of these do increase anyway. So you could use either one of these. But as you can see, as the volume or surface area increases, the surface area to volume ratio decreases. That's what we have to do. So as size increases, the surface area to volume ratio, again, you can't write in shorthand like I'm doing right there. You can't write SA to V, but hopefully you see, decreases. So you do have to write it in like full words. You can't use random acronyms unless I specifically say so. We have here organism B exchanges gases with the environment directly through its skin. Organism D exchanges gases with the environment using respiratory system instead. So B is most likely some kind of insect and D is a larger animal. Explain why organism D requires a respiratory system but B does not require a respiratory system. Well, again, think about B being an insect, D being a larger animal like a cat. That might help. It's all to do with the size of the organism. So if we have a look at B and D, B is over here, D is over here. As you can see, what is the difference between these two? Well, D is of course bigger, right? That's the only difference between B and D that we can tell because they've given us no other information. So the only difference between them is one is bigger than the other. So why does that matter? Well, D has a smaller surface area to volume ratio. So what does that mean? Well, because it's bigger, it means that when you take its surface area and divide it by its volume, it's a smaller value. This means that it can't get sufficient gas exchange through the surface of its skin, and it needs instead a respiratory system. So let's word that in terms of biology. Well, D has smaller surface area to volume ratio. Again, you'd want to write that in words. Ratio than B. Now, the real reason that insects can breathe through their skin, so can certain types of frog as well, I believe, and big organisms can't, is because the diffusion distance is too long for sufficient diffusion to take place. So, this means that organism D has a much larger diffusion distance than B. Diffusion cannot provide enough. Again, I'm going to assume it's oxygen. Let's say gas exchange, because technically speaking, it might be a plant. So yeah, enough gas exchange to meet the needs of the organism. That's probably the way I would write it. So D is a smaller surface area to volume ratio. This means the diffusion distance is too big, essentially, for you to have a system where you breathe directly through your skin. The next question, they've repeated the same table, so we don't need to go through the table again, although it's kind of annoying. I mean, no, it makes sense they haven't put X in there, actually. But for these two organisms, we have a metabolic rate as well. So D and E, if you look, that means D. Again, this is just what I would do in the exam. E is the bigger one, right? Because there's a reason why they picked D and E. So, they both need to maintain a constant body temperature, of course. That means they're warm-blooded. So why is the metabolic rate of D bigger than E? And it says, again, use the information in the tables. Well, again, think about why they've done that. So what is the difference between D and E? Well, D is smaller than E. Now, smaller things dissipate heat a lot faster because of this surface area to volume ratio. So it's once again using the same type of thing. So D has a larger SA to volume ratio and will lose heat faster than E. So what? So we've established that because there's a larger surface area to volume ratio, it will lose heat faster. That's the key thing. Again, smaller things lose heat faster. That's why almost everything you find in a desert is small, like desert rat, desert mice, scorpions, spiders. Whereas everything in very cold climates are a lot bigger. Penguins, polar bears, seals, walruses, things like that. Because, again, you want a really low surface area to volume ratio if you're in a cold climate. So, the way that small animals compensate for this is they have a much higher metabolic rate. Because metabolism releases heat, so it keeps you warm. So D requires higher metabolic rate. I know I spelled that wrong. It's meant to be one L. As heat is released from respiration. As heat is released from respiration, and more heat is needed to keep D warm. That's the way I'd write it. So D is a large surface area to volume ratio and will lose heat faster than E. This means D requires a high metabolic rate as heat is released from respiration, and more heat is needed to keep the organism warm. In my opinion, one of the best questions you can get in your paper 1. So I've already seen it says alveoli and villi. I know exactly it's going to talk about some diffusion. So we have alveoli in the lungs and villi in the small intestines. So how are they both adapted to increase absorption? Well, luckily for us, they're adapted in the exact same way. Maximize surface area, very thin, good blood supply. That's all we basically need to talk about. So, both have a large surface area, but again, remember, we need to put 4 points, but we need to give justifications for all of them. So both have large surface areas, so what? To maximize diffusion. That's simply directly to maximize diffusion, so that's one done. What about next? Both have thin walls. They tend to only be 1 cell thick. And what does this do? It reduces the diffusion length. You can call it diffusion distance or time if you want. I'll leave it as distance for now, again, so that's 2 marks already. I'm just going to leave it as both. Both have good blood supply, as you can see from the capillary networks around them. Why is that useful? Well, again, to maintain a concentration gradient, or, well, I mean, actually, let's do that for the next one. Reduces, again, diffusion distance, because again, the reason why they're close is because they're very thin walls. Both supplied by, I mean, another thing you could say is both have good by capillary network. I'm actually going to, instead of repeating myself, I'm going to write the next one here. This would be an extra point as well, but this also means it maintains a concentration gradient. So the idea here is, if you're having this exchange of materials, so to speak, so gases or minerals or whatever, you don't want the concentration gradient to dip. You want there to be a constant big gap, so that way you maximize the amount of diffusion you have. Increase absorption. What else could you say? Well, you could say that the cells have, in the villi, not in the alveoli, but they have many mitochondria to provide energy for active transport. And you could also talk about the fact that they have microoli in the villi to increase surface area further. But these are the ones that affect both of them, and so they're probably the easiest to write about. But the idea is having a good blood supply reduces diffusion distance, maintains concentration gradient, and so on. Perfect, let's move on to the, I believe, last question. So this is a pretty hefty topic. It has quite a lot of writing in it and quite a lot to remember. So I'm going to try and go through it as best I can. But again, remember with biology, a lot of it is memory. So I'm trying to explain things and relate them to analogies to help you remember. But again, when in doubt, those good old-fashioned flashcards are pretty good. So we have HIV is a pathogen. Give one way HIV can spread. Hopefully this is a bit general knowledge, but HIV is sexually transmitted. So you can just say sexual intercourse. So why does it say one way? Well, there is, I should say, unprotected sexual intercourse, technically. There is another way you can get HIV, and again, you'd only need one of these, but the other way is an exchange of bodily fluids. So, for example, if your blood mixes with someone with HIV, you can get HIV. You can also get it from mother to a unborn baby as well, through the, I was about to say fallopian tubes, not through that, through just the transmission of minerals and fluids and stuff, sharing needles, blood transfusions, pretty much anything like that. Yeah, so basically it's a sexually transmitted disease. It's probably the easiest one to do, rather than talk about any of the other ones. This next kind of trend question is a bit interesting. So we have this table, number of HIV cases in women and men, and as you can see, it kind of goes down, up, down, down, and here it goes up, down. So describing the trends, you basically just say what I just said. It's a bit weird, so I would break it up into 2 bullet points. In women, I would try and use an interesting word. I would say it fluctuates. So it's going like down and up and down or whatever, but you can just literally say cases decreases, increases, then decreases. Just so you're aware, I did actually, before putting on the recorder, I double checked this one because I was like, okay, could I just say it fluctuates? And the mark scheme does say you can say fluctuates, but it does literally say this: it decreases, increases, then decreases. Very weird, because that's not really a trend. A trend normally is decrease, increase, or it fluctuates, or there is no trend. But just so you're aware for 2024, just in case something like this comes up, when they say describe the trend, you're literally talking about year to year what happens. Well, it decreases, but it doesn't keep decreasing. It does increase, and then it decreases again. So you do need to mention that. And then in men, it's a similar type of thing. The cases increase, then decrease. and that's completely fine. And that's it. Hopefully not too shocking. Bit of a weird wording, but that is what they want. For part 3, so just one reason for the change in the number of new HIV cases between 2014 and 2018. So I like to highlight these things. So if you'd like, you're looking at this, well, what's happening? Well, this is going down, this is going down. So basically, explain how or describe how these number of cases could go down. So again, this is probably something, there's so many different answers to this. So I'm just going to start going through it. 1, better education. So maybe sex education got better in schools, so people are more aware and so they're more protected. So better education about it to prevent the spread. Perhaps condoms are more easily accessible. That's another thing. New or better drugs to help prevent HIV or spreading HIV, so being able to treat it. And better testing. So if people are tested and identified, then it prevents the spread. So just so you're aware, is someone could have HIV, or at least back in the old days, they could have HIV, but then in a test it shows up negative. So they're saying, oh no, you don't have HIV. And then they go, have sex with a bunch of people or do heroin or something, and they spread it. Which is horrifying, but that can happen. But now, because testing is a lot better, people get tested and it's more likely to be accurate. So instead of being, let's say, 90% accurate, it might be 99.9% accurate. This means that the people that have HIV know it, and therefore they're more able not to spread it. So any one of those reasons, I'm not going to write anything else. Some of the things you can't say, by the way, is you can't say something like vaccination because there is no vaccine for HIV, and you can't say less promiscuity. So there's an idea that people are having less sex. Although there are studies to suggest that, it hasn't been linked to HIV, is basically what they're saying, so you can't really say anything like that. In general, try and keep anything somewhat related to politics out of science. Calculate the ratio of new cases in women to new cases in men in 2018. Cool. So what you want to do is take the number of cases in women divided by the number of cases in men. So in 2018, right? So 242 over 1288. It's interesting how much bigger the number of cases in men are than women. I mean, you'd think it'd be roughly even considering... Yeah, who knows. Anyways, so go through that, divide it, and you're done. Over 1288. And it says give your answer to 3SF. Look at this, it's a maths question basically. 0.188. That's 3 marks. Like I said, if you're halfway decent at maths, you can get quite a few nice juicy marks in your biology exam. So in the UK, the UK population, the total number of women is greater than the total number of men. The data in the table is used to compare the proportions of new cases. So how could it be presented differently? In general, and this works for, by the way, this is a life thing, and just be aware of data in real life, we almost always give it as percentages because of this very reason. It doesn't make sense to look at this and say, there's a greater proportion of men than women getting HIV, because what if there's 10 times the number of men than women in a country? Right? You don't know. So instead, it's better to give it as a percentage of the total population because it removes the fact that there's a different number of men and women. Completely nullifies that effect. So generally speaking, if they ever ask this, the best thing to do is give as a percentage. And that, if you notice in real life, in a lot of data on the news or on the internet, numbers are given as percentages because it removes the discrepancies in the population. The other thing you could say is give the numbers as per 100,000, which, by the way, is basically the same as giving it as a percentage, because it's just per 100 people, but let's leave it as that. So scientists have been working to produce a HIV vaccine for many years. Explain how a vaccine could work. So, by the way, this is a bit of a red herring question because it's not really about HIV. It's about how vaccines work. That's actually the question you're answering. How do vaccines work? So, let's get out our vaccine-related knowledge. So what happens? Well, and this, by the way, is the key thing, is it's how vaccines work, but replace the word pathogen with HIV. So, for example, inactive pathogen is injected into the body, becomes inactive HIV injected into the body. That's it. So inactive, you can also say weakened or whatever. Inactive HIV injected into the body. Remember, HIV stands for human immunodeficiency virus, so you don't need to say HIV virus, although a lot of people do say that in real life, into the body. Boom, that's one mark. White blood cells produce antibodies against HIV. That's 2 marks. Again, this is just, normally you'd say white blood cells produce antibodies against pathogen, but because we have a named virus, we can call it that. If it was for measles, you'd say the same thing but replace HIV with measles. If it was rubella, you can replace it with that as well. Okay, so then if person becomes infected with HIV at some point in their life, the specific antibodies are rapidly produced, which means the antibodies destroy the virus before a person gets sick. Because the problem with HIV, and the reason why you don't really get better from it, is because it targets your immune system. So it gradually weakens your immune system to the point where it never ends up actually developing resistance to it. However, if you could preempt the resistance, that could work if we can get a vaccine for it. So once again, this question is speaking about HIV, but it's a question about how vaccines are made. So this is how vaccines are used. This is how they work. This is how they are made. So monoclonal antibodies. Again, this is a standard 4 marker. Just for some context, this whole thing about HIV developing AIDS is assisted immunodeficiency syndrome. Basically, someone's immune system becomes so weak that even common illnesses and diseases, so all around you right now is bacteria that your body manages to fight off before you even get sick at all, that becomes deadly, which is why these people need to sometimes be kept in complete clean rooms. So I'm not even talking about the cold virus, I'm talking about viruses that already exist all around us anyway, that we never even get sick from. Those kinds of viruses can kill these people. So how is this made? Well, the HIV, you can also say antigen or protein. So normally, again, in a normal case, you would say antigen. But because we're talking about HIV specifically, I'm going to remove the word antigen and use HIV. In the mark scheme, you can use either. So don't worry. Is injected into a mouse. 1 mark. You extract mouse's lymphocytes that are making the antibody to HIV. So again, the idea is you inject HIV into a mouse. The mouse's lymphocytes, their white blood cells, produce the antibodies. You extract these white blood cells and combine the lymphocytes with tumor cells. So why is this really useful? Well, tumors are cancer, right? Cancer's really useful because what cancer is this uncontrollable cell division. That's why they grow so quickly. Because instead of having the normal limit, they just keep growing forever. So the idea here is you're going to produce a hell of a lot of lymphocytes in a short period of time. Creating something called a hybridoma. This hybridoma is cloned to create many cells that produce the antibody. And the idea then is you have a bunch of this antibody which you can give to other people as a vaccine. And again, this monoclonal because it all comes from the same DNA, right? It comes all from this one mouse. Lastly, but certainly not least, we have one more part. Not gonna lie, I forgot about this part. I thought this might be the end. This shows how HIV enters a human cell. We have the HIV antigen bonds to a human cell. HIV antigen bonding site injects in. Again, this is basically how every single virus on the planet works. Bonds and injects its DNA into your cell. Your cell then starts producing more of the virus. explodes out of the cell, and repeat process until you die or you fight it off. So it's just how the monoclonal antibody for HIV helps to prevent a person infected with HIV developing AIDS. Well, again, this is 3 marks, so let's do 3 quickie little bullet points. The monoclonal DNA, the monoclonal antibody, is specific to HIV antigen. Okay? So this monoclonal antibody will only bind to the HIV. That's important, because otherwise it would attack any cell. Good. These antibodies bind or attach to the HIV antigens. Cool. This means, so again, we have these antibodies, they bond here, it can now no longer bond to the human cell. So you can imagine it as you can see here, one of these antigens bonds to these bonding sites and they go in. However, imagine if you had a cap on all of them. Well, now it can't bond because you've basically gotten rid of them. Something's already bonded to it. It's like, imagine if, the best way I can think of it is the lock and key diagram, right? If I put my key into my front door, you can't then put your key into the front door as well, right? Because it's already taken up. In the same way that if an antibody bonds to one of these sites, one of these antigens, that antigen can't bond anywhere else because it's already stuck to something. Second mark. This means the HIV genetic material, it's not DNA, I was about to say DNA, but it's not DNA, cannot enter the human cell. Again, do not say DNA because it's a virus. Viruses do not have DNA. And that does bring us to the end of the paper. I hope you found that useful. If you did, please leave a comment just saying if you found it useful or not, because I will do more of these if possible. If there's any topics you might want me to go through, let me know. But I might do some more of these papers depending on what. I'm probably going to do one for chemistry and physics, just see how people like it. If people don't, that's totally fine. I'll stick to maths. I'm not too bothered. But I just wanted to see if this would be helpful to some of you guys. So again, do let me know, but I'll see you in the next one.